Hi guys, and welcome to this session of NEET biology practice. So we are taking another past paper and discussing the questions from there. So let us move ahead with the first question. Okay, so the question over here is a non-proteinaceous enzyme is. Okay, now we are talking about an enzyme. Okay, so when we are talking about an enzyme, let us first remember that ASE is the suffix that denotes an enzyme, correct? Okay, but that is the suffix that is generally used for proteinaceous enzymes. So ligase and deoxyribonuclease. So these are proteinaceous enzymes that we are seeing in here. Lysozyme is actually another proteinaceous enzyme which degrades the bacterial cell wall. Okay, ribozyme is a non-proteinaceous enzyme. Why? Because ribozyme is nothing but RNA. Okay, so ribozyme is an RNA molecule that acts as an enzyme. Yep. Okay, so ribozyme is the correct answer over here. Now select the wrong statement. They say bacterial cell wall is made up of peptidoglycan. Is it? Yes, it is. Recall that bacteria have something which is called as the cell envelope. Okay, and in this cell envelope what we have is the outer glycocalyx. Okay, then inner to the glycocalyx what we have is the cell wall. Okay, and inner to the cell wall what we have is the cell membrane, isn't it? Yep. Now the glycocalyx is made up of polysaccharides, cell wall is made up of peptidoglycan, and the cell membrane is made up of phospholipids. Yeah, cell membrane is made up of what? Phospholipids. Got it? So bacterial cell wall is made up of peptidoglycan. Pili and fimbriae are involved in motility of bacterial cells. Is that true? We have to select the wrong statement. That is wrong, isn't it? Why? Because motility of bacterial cells is because of flagella. Then what are pili and fimbriae involved in? Pili are involved in something which is called as conjugation. Okay, what is conjugation? Conjugation is a primitive type of sexual reproduction between bacteria where transfer of plasmid takes place. So if there is a bacterium, so if this is basically a bacterium that has a plasmid, so this is the DNA, okay, and here this is the plasmid. Okay, now if we have another bacterium that does not have the plasmid, then the pili of this bacterium and pili of this bacterium, they will form something which is called as a conjugation bridge, and through this pillars the plasmid will be transferred to the other bacterium. Okay, and then this bacterium can form conjugation bridge with another bacterium and provide the plasmid to it. So that is what is conjugation, and pili are involved in that. Fimbriae are adhesion structures, so they help the bacteria stick or cling onto something. Okay, so that's the function of the fimbriae. So pili and fimbriae. So pili and fimbriae are mainly involved in motility of bacterial cells is wrong. Yep, they have their functions are different. Okay, next, which of the following biomolecules is common to respiration mediated breakdown of fats, carbohydrates and proteins? Yep, respiration mediated breakdown. Okay, now when we talk about respiration mediated breakdown, see, let's think over it. So we have respiration, and respiration we have aerobic and anaerobic respiration, isn't it? Yeah, aerobic and anaerobic respiration. When we talk about aerobic respiration, so we know that glucose undergoes glycolysis. Yep, where the first molecule is glucose 6-phosphate. So glucose undergoes phosphorylation and forms glucose 6-phosphate. Then it changes to fructose 6-phosphate and then fructose 1,6-bisphosphate, so on and so forth. So glucose 6-phosphate and fructose 1,6-bisphosphate are actually a part of what? They are a part of glycolysis pathway. And so is pyruvic acid, isn't it? So pyruvic acid is the one that is formed at the end of glycolysis. Yeah. But acetyl-CoA. Now when we talk about acetyl-CoA is seen in link reactions and it is also seen in Krebs cycle. Okay. It is also seen in oxidation of fats and proteins, where it is again, where the compounds are again broken down and converted to acetyl-CoA, and then the acetyl-CoA enters the mitochondria and the Krebs cycle. Got it? Yep, that's basically how acetyl-CoA plays an important part in breakdown of fats, carbohydrates and proteins. Okay, so the answer over here will be what? Acetyl-CoA. Next, you are given a tissue with its potential for differentiation in an artificial culture. Which of the following pairs of hormones would you add to the medium to secure shoots as well as root? Yep. Now, indole acetic acid is what? It's an auxin, right? Indole 3-acetic acid is an auxin. And then we have gibberellins. Do gibberellins, are gibberellins responsible for root formation, shoot formation anywhere? They are not. So wherever we have gibberellins, that option is gone straight up. So here is gibberellin, here is gibberellin. Gibberellins are not involved. Gibberellins are involved in stem elongation, right? Then abscisic acid. We see abscisic acid in 2 more options. What is abscisic acid responsible for? Abscisic acid is responsible for abscission, that is the falling of leaves, isn't it? Yeah. So is that a method of root formation and shoot formation? No, it's not a method of root formation and shoot formation. So there is abscisic acid is absolutely out of the question now. Got it? So auxin and cytokinins ultimately becomes the correct option. Why? Because auxin is responsible for something which is called as rhizogenesis. Rhizogenesis is what? Root formation. Yeah, rhizogenesis is what? Root formation. And then we have cytokinins, which are responsible for collagenesis, that is shoot formation. Yeah. So auxins and cytokinins, if they are added to the nutrient medium in tissue culture, what will happen? You will have an undifferentiated mass of cells. If you treat them with auxins and cytokinins, they will start producing roots and shoots respectively. Okay. Next question, what we have is the process which makes major difference between C3 and C4 plants. Glycolysis. Now glycolysis, we just discussed that it is a pathway of respiration where glucose is broken down. Okay, it's a respiratory pathway. Calvin cycle. Calvin cycle is literally the C3 cycle, isn't it? Yep. So C3 cycle is taking place in C3 plants, obviously. C4 cycle also sees Calvin cycle where? In the bundle sheath cells. Yeah. Are you getting it? Where do we see them? We see them in the bundle sheath cells. So C3 cycle is also seen in the C4 plants, but in the bundle sheath cells. Why are they called as C4 plants? Because the first stable compound is oxaloacetic acid. Yeah, which is a 4-carbon compound. So the initial acceptor is what? Phosphoenol pyruvic acid, which is a 3-carbon compound that takes up CO2 and forms oxaloacetic acid. So this is what happens in C4 pathway. But later again C3 pathway takes place where the carbon dioxide enters the C3 cycle in the bundle sheath cells. So respiration is another aspect that is common to all the plants because that is how energy is produced, isn't it? Then what is the difference is photorespiration. Now photorespiration is a process that is detrimental to C4 plants. Are you understanding? Photorespiration is detrimental to C4 plants, but in C3 plants, photorespiration, small amount of photorespiration is beneficial. Why? Because it prevents the photooxidation of chlorophyll A. Okay, but in C4 plants, if the C4 plants undergo photorespiration, they will lose already fixed carbon dioxide. So the major difference between C3 and C4 plants is what? Photorespiration. Are you getting it? Yep, simple enough. So there's photorespiration in these plants. Next, photorespiration is the difference. So for C3 plants, it is beneficial. For C4 plants, it is detrimental. Next, what we have is the ovule of an angiosperm is technically equivalent to. So let us look at the angiospermic structure. So recall that the flower is born on the thalamus, isn't it? Yep. And thalamus is nothing but condensed internodes. So from every node here, what you get is a structure, is a part of the flower. So flower is nothing but a modified shoot. So whatever components you are seeing with the flower, they also carry a term which is called as fill, right? Which means leaf, correct? Fill means leaf. So when we talk about the flower, okay, when we talk about this particular structure, what is this? This is stigma, style and ovary, right? This is the carpel. Now carpel in technical terms is known as what? Megasporophyll. So it is a leaf-like structure that bears the megaspore. Okay, megaspore. It bears the megaspore. But what is the megaspore? Megaspore is the female gametophyte. Yeah. What is megaspore? Megaspore is the female gametophyte or the embryo sac. Now this female gametophyte or the embryo sac is found where? It is found in this particular structure which is known as what? The ovule. Correct? So this is the ovule. And this is the embryo sac. Are you getting it? That is the ovule and that is the embryo sac. Now embryo sac is present inside the ovule. So embryo sac is the megaspore. Megaspore is present inside the ovule. So the ovule is the one that is bearing the megaspore. So the ovule becomes the megasporangium. Right? Ovule becomes what? It becomes the megasporangium. What is megaspore mother cell? Megaspore mother cell is the cell inside the ovule which is going to develop into the embryo sac by meiosis and then subsequent mitosis. Okay. Next, the mechanism that causes a gene to move from one linkage group to another is called. Now here we need to understand what are linkage groups first and then we will move ahead to whatever is given over here. Linkage groups. Recall Morgan's experiment. Okay, Morgan gave us the idea of linkage. He gave the phenomenon of linkage. So when we talk about linkage are of two types. So we have complete linkage and incomplete linkage. Now what does it mean? It means that the genes that are located in this complete linkage, genes are located very close to each other. Yeah, genes, where are genes? Genes are very close to each other. Okay, the completed genes are very close to each other because of which they do not separate during crossing over or meiosis. Are you getting it? They do not separate during crossing over or meiosis. In incomplete linkage, the genes are located far from each other. So genes are away. Yeah. Because of which they can separate during meiosis. Okay, during crossing over they can separate out. But when we talk about genes on a chromosome, so if this is a chromosome, this is literally a segment of DNA which has been arranged as the chromosome and has many genes in it, isn't it? Genes on it. So all these genes which are a part of the same chromosome, yeah, all these genes which are a part of the same chromosome can be said to be linked to each other. Now they can be completely linked or incompletely linked based on the position. So if I write A, B, C, D, E, F, G, H, so on and so forth. So A is also linked to H because it is part of the same chromosome. But since they are away from each other, they will be termed as what? Incompletely linked genes. But G and H are very close to each other, so they will show complete linkage. Simple enough, okay? So every chromosome forms what? A linkage group, right? Every chromosome forms what? A linkage group. So how many chromosomes do we have in total? We have 46 chromosomes in total, but does that mean we have 46 linkage groups? No. 46 are actually what? The diploid number, isn't it? And every chromosome is present as another copy, right? So homologous chromosomes. So linkage group is equal to the haploid number of chromosomes. Yeah. So if we look at the haploid number, how many linkage groups should we have? 23. But 23 is for female. Okay. For male, it will be 24. Why? Because in the female, the XX chromosome, the sex chromosome complement is homologous. Whereas in the male, X and Y are not homologous chromosomes. They are two independent chromosomes. So in male, we will have how many? In male, we will have 24 linkage groups. So if it says the mechanism that causes a gene to move from one linkage group to the other, that means what? From one chromosome, like if this is chromosome number 1 and if this is chromosome number 5, yep, so chromosome number 1 is one linkage group, chromosome number 5 is another linkage group. So the gene should be moving from chromosome number 1 to chromosome number 5 or vice versa. That can happen between any chromosomes. So what can this be termed as? Let's take a look at chromosomal aberrations. So when we talk about inversion, what happens over here is, let's say this particular segment, okay? So if I have genes G, H, I, J over here, in inversion, what is going to happen? One of these pieces is going to get cut out and it is going to get inverted and inserted again. So G, I, H, J. So I and H got inverted, but did that happen on another chromosome? No, it happened in the same chromosome. Duplication will be simply the addition of more genes by duplication. So GH, GH, G, H, I, J. So can you see that G and H got duplicated and added again to the chromosome? Yep. Crossing over is the exchange of genes between homologous chromosomes. Here it says one linkage group to the other. Yep. So it is not happening between two linkage groups. It's happening between homologous chromosomes. So what is the answer over here? It is translocation. Okay. So in translocation, literally one part of the one chromosome will get transferred to another chromosome. Yeah. That is what is translocation. So, excuse me, remember that translocation occurs between chromosomes that are non-homologous. So non-homologous chromosomes are going to have translocation, and the non-homologous chromosomes are also going to show you movement from one linkage group to the other. Yep. If it is happening crossing over, then crossing over is always between the homologous chromosomes. Yep. Next, the equivalent of a structural gene is muton, cistron, operon, recon. Okay, let's understand this. This is the gene. Okay, so here when we talk about units, so these are genetic units. So muton is a unit of mutation, right? Muton is the smallest unit of mutation. So it can be even one nucleotide. Are you getting it? Muton, a unit of mutation, is it can even be one nucleotide, because there can be addition mutations, deletion mutations, transition mutations, transversion mutations. So these mutations are happening with a single nucleotide, so point mutations. So muton is a unit of mutation. Recon is a unit of recombination. So if we are talking about recombination, these are the genes that undergo recombination or exchange between homologous chromosomes. Are you getting it? They undergo exchange between the homologous chromosomes. So that is unit of recombination. Are you getting it? Yep. So that is recon. Operon is what? Operon is a cluster of genes, right? Operon is a cluster of genes which is seen in prokaryotes, and they have a particular function, a common function. Okay, so operon is a cluster of genes that has a common function. Got it? So this is basically what operon is. Cistron, okay, is the unit of expression. So when we say a gene has expressed and given us an mRNA, and this mRNA has given us a polypeptide, yeah, has given us a polypeptide, then that is what is known as what? Cistron. So unit of expression is a cistron. Got it? So structural gene is nothing but cistron. Yeah. One thing that we need to know, that eukaryotes, when we look at a transcription unit, in transcription unit we have a promoter and a terminator. Okay, we have a promoter and a terminator. Now in prokaryotes, between a promoter and a terminator, we may have more than one structural genes. And this condition is known as polycistronic. Yeah, this condition is known as what? Polycistronic. In eukaryotes, however, between a promoter and a terminator, there will be only one structural gene. And such a condition is known as what? Monocistronic. Yeah. So remember that the genes of prokaryotes are polycistronic and the genes of eukaryotes are monocistronic. A foreign DNA and a plasmid cut by the same restriction endonuclease can be joined to a recombinant plasmid using what? What are these enzymes? This is a restriction endonuclease, isn't it? Yeah. Taq polymerase is literally a polymerase which is utilized in DNA synthesis. But where do we see Taq polymerase? Taq polymerase is seen in, we use Taq polymerase in PCR, isn't it? It comes from the bacterium Thermus aquaticus. Why? Because Thermus aquaticus is a thermophilic bacterium and its polymerase is heat stable. It is heat stable, it is heat dependent. So not heat dependent, it is heat stable, because of which it can withstand the high temperatures of PCR. Okay, it can withstand the high temperatures of what? PCR cycling, and that is the reason Taq polymerase is used in PCR, polymerase chain reaction. DNA polymerase 3 is another polymerase, isn't it? So it is also going to be utilized in what? Synthesis of DNA. Yeah. A foreign DNA and a plasmid cut by the same restriction enzyme can be joined, they are asking. So who does that? It's ligase, isn't it? Ligase will be doing that. Why? Because ligase, what does it do? Ligase creates phosphodiester linkages. Yeah, ligase creates phosphodiester linkages. So EcoR1 is a restriction endonuclease. So if we have a DNA molecule, if we have this particular DNA molecule, EcoR1 will recognize a sequence called GAATTC. Okay, GAATTC, 5 prime to 3 prime, 3 prime to 5 prime. This is the restriction site. So what will happen over here is, this is how it will cut it. So we will get sticky ends. Okay, and these sticky ends from the two DNA sources will be joined by what? Ligase. So a foreign DNA and plasmid cut by the same restriction endonuclease can be joined to a recombinant plasmid using ligase. Yep, that is the answer. What kind of therapy was given in 1990 to a 4-year-old girl with adenosine diaminase deficiency? Adenosine diaminase is an enzyme that is required for the functioning, the proper functioning of T lymphocytes. Okay, it is required for the proper functioning of T lymphocytes. So adenosine diaminase. So T lymphocytes maturation, so that they can provide you with immunity, cell-mediated immunity, it involves adenosine diaminase functioning. Now, the failure of adenosine diaminase causes something which is called as severe combined immunodeficiency disorder, or SCID. Yeah, it causes what? Severe combined immunodeficiency disorder, or SCID. So what was done over here? A functional gene of adenosine diaminase, okay, adenosine diaminase functional gene was taken, transferred to T lymphocytes with the help of retroviral vectors. Okay, transferred to T lymphocytes with the help of what? Retroviral vectors. And these retroviral vectors, I mean, when the gene was transferred to the retroviral vectors, they were allowed to infect the T lymphocytes. And these T lymphocytes carrying the new functional ADA gene, adenosine diaminase gene, were transfused to the patient. So this was the first successful application of gene therapy, okay, where a girl called Ashanti De Silva was provided with gene therapy for SCID, severe combined immunodeficiency disorder. Yeah, chemotherapy, radiation therapy, immunotherapy, cancer treatments. Next, if plus sign is assigned to beneficial interaction, take it negative sign to detrimental, and 0 sign to neutral interaction, then the population interaction represented by plus and minus refers to what? Mutualism is where, see, you have species A, species B. In mutualism, both are benefiting from each other, correct? Both are benefiting from each other. So hence, both will have the plus sign. Amensalism and commensalism. So amensalism is a condition where one is having a detrimental effect, the other is neutral. In commensalism, one is having a positive effect while the other is neutral. Whereas parasitism is the condition where species A gets positive interaction at the cost of species B. So a parasite feeds on a host, okay? So the parasite gets the nutrition or the benefits by taking it from the host. So hence parasitism is what will be represented by the plus and minus sign, okay, parasitism. So this is how different population interactions will be noted as, okay. Which of the following sets of diseases is caused by bacteria? Okay, so let's just look at it. So herpes, influenza. Herpes is a viral disease, okay. Influenza can be viral or it can be caused by a bacterium called Haemophilus influenzae, okay. Tetanus is caused by a bacterium called Clostridium tetani. Mumps is caused by a virus, so it's a viral disease. Typhoid is caused by Salmonella typhi, it's a bacterial disease. Smallpox is caused by a virus, smallpox virus, so it's a viral disease. Cholera, tetanus. Cholera is caused by a bacterium called Vibrio cholerae, okay, and tetanus is caused by Clostridium tetani. Both of which are bacteria, okay. Tetanus is caused by Clostridium tetani, both of which are bacteria. So which of the following sets of diseases is caused by bacteria? Is cholera and tetanus, okay. Next, oxidative phosphorylation is formation of ATP by transfer of phosphate group from a substrate to ADP. The option literally says substrate to ADP. So such a reaction will be termed as what? Substrate level phosphorylation. Yep. It will be termed as what? Substrate level phosphorylation. Oxidation of phosphate group in ATP. That's literally breakdown, isn't it? Yep. Addition of phosphate group to ATP is phosphorylation, that is correct. But which kind? It's just plain, it just says plain addition of phosphate group to ATP. We don't know what they are specifying over here. Formation of ATP by energy released from the electrons removed during substrate oxidation. Yep. This is what is oxidative phosphorylation. So where does this happen? This happens on the inner mitochondrial membrane. Yeah, where does this happen? This happens on the inner mitochondrial membrane, and on the inner mitochondrial membrane, okay, on the inner mitochondrial membrane, what we have over here is, on the inner mitochondrial membrane, we have the coenzymes. Yeah, what do we have? We have coenzymes. And what are these coenzymes? These coenzymes are NADH, okay, NADH2, FADH2, okay, then we have ubiquinone, then we have cytochromes, okay, and then we have oxygen. Yep, half O2. These are electron carriers. So what happens over here is, these components, they take electrons and they give electrons. They take electrons and they give electrons. So they get alternately oxidized and reduced. Okay, they alternately get oxidized and reduced alternately as they transfer the electrons. And as they transfer these electrons, the energy of the electrons is utilized for formation of ATP. So this, since this involves oxidation and the transfer of energy for ATP formation, this is known as what? Oxidative phosphorylation. And the progression is known as electron transport chain. The progression is known as what? Electron transport chain. Correct? So that is what is oxidative phosphorylation. Which of the following is the least likely to be involved in stabilizing the three-dimensional folding of most proteins? Hydrogen bonds, though we know, are definitely there. Okay, for example, even when the proteins undergo alpha helical formation, there are hydrogen bonds dropped from every 3.4th amino acid, isn't it, which stabilize the helix. Then we have beta pleated sheets. Okay, yeah, which again stabilize the sheet. Fine. Now when these move on to these are secondary structures, when they move on to tertiary and quaternary structural formations, what happens is you have one protein. Okay, it may be alpha helix or beta helix or both of them, tertiary compound, and when they join together, there are something which are called as hydrophobic interactions. Okay, these hydrophobic interactions are majorly disulfide bridges. Okay, so we have hydrogen bonds that stabilize the interactions. We have electrostatic interactions, salt bridges, that stabilize the structure, and hydrophobic interactions that stabilize the structure. The question asks which of the following is least likely to be involved in stabilizing the three-dimensional folding of proteins. So that is ester bonds. Okay, so disulfide bridges will be formed with an amino acid called cysteine. Yeah, and there will be other sulfur containing amino acids which will also be utilized for hydrophobic interactions, which are disulfide bonds. And these disulfide bonds are very strong components and they stabilize the quaternary structure to a very great extent. Next, what we have is which of the hormones stimulate the secretion of pancreatic juice and bicarbonates? Okay, angiotensin is involved in renin-angiotensin mechanism, which is responsible for what? Increasing the blood pressure, for increasing the glomerular filtrate rate, isn't it? GFR, for increasing GFR. So angiotensin is definitely not the answer. Epinephrine is nothing but adrenaline. What is its function? Adrenaline is an emergency hormone, right? Next, gastrin and insulin. Insulin is what? Insulin is the hormone that is responsible for lowering the blood glucose level, isn't it? So insulin is the hormone that lowers the blood glucose level. Gastrin is the hormone that tells the gastric glands to secrete gastric juice. Cholecystokinin and secretin are actually the ones that are responsible for this particular function. So secretion of pancreatic juice and bicarbonates. So cholecystokinin tells the liver to produce bile and also causes the secretion of bile from the gallbladder, which contains bicarbonate ions. Secretin stimulates the pancreas. Okay, secretin stimulates the pancreas to release pancreatic juice. Okay, to release what? Pancreatic juice and bicarbonates from pancreas. Insulin and glucagon. Insulin we just discussed. Glucagon is a hyperglycemic hormone. So what is it going to do? It is going to bring about the breakdown of glycogen, okay, and release of glucose into the blood. Got it? Next, the posterior pituitary gland is not a true endocrine gland because it is provided with a duct. Is it? No, it's a ductless gland. Okay, it only stores and releases hormones. Now that is the answer over here. Why? Because pituitary gland has two parts. It has the anterior pituitary, anterior part, and the posterior part. Anterior part is known as adenohypophysis, and posterior is known as neurohypophysis. Yeah, posterior is known as what? Neurohypophysis, adenohypophysis and neurohypophysis. Now, adenohypophysis, neurohypophysis, adenohypophysis is the actual endocrine structure which secretes the hormones. Okay, posterior, which is neurohypophysis, it is in connection with the hypothalamus. Okay, so it receives secretions from the hypothalamus. So hypothalamus synthesizes antidiuretic hormone, okay, and oxytocin. ADH and oxytocin are synthesized by the hypothalamus, and the hypothalamus sends these hormones to the pituitary, where the pituitary stores them and releases them when signaled by the hypothalamus. So pituitary, which pituitary? The posterior pituitary. So the neurohypophysis or the posterior pituitary is not a true endocrine gland because it does not synthesize its own hormones. It receives the hormones from the hypothalamus and secretes them into the bloodstream when stimulated to do so. Okay. Next, the part of nephron involved in active reabsorption of sodium is descending limb of the loop of descending limb of Henle's loop. Distal convoluted tubule is involved majorly in what? It is involved in tubular secretion. Okay, it is involved in tubular secretion, and especially it secretes potassium ions. Now, why does it secrete potassium ions? It secretes potassium ions to balance out the positive charges in the glomerular filtrate. Okay, because from the glomerular filtrate a lot of sodium has been reabsorbed. So to maintain the osmolarity of the filtrate, the distal convoluted tubule secretes potassium into the filtrate as the filtrate passes through it. Proximal convoluted tubule is majorly responsible for what? It is responsible for absorption of a lot of things: glucose, amino acids, then water, etc. Bowman's capsule is only the part where the glomerulus is present, and through the glomerulus the filtrate moves to Bowman's capsule, podocytes, etc., and causes what? Ultrafiltration. Okay, so the part of nephron involved in active reabsorption of sodium is nothing but descending limb of Henle's loop. Embryo with more than 16 blastomeres formed due to in vitro fertilization is transferred into what? So in vitro fertilization, you have the ovum, and with the help of a micro injection, sperm is injected into the ovum through a process called as intracytoplasmic sperm injection, or ICSI. Yeah, intracytoplasmic sperm injection, or ICSI. Now this intracytoplasmic sperm injection, or ICSI, results in what? Fertilization, isn't it? Yeah, what does it result into? It results into fertilization. Now when it results into fertilization, it will now form the zygote, and the zygote will undergo embryo formation and form the embryo, the blastocyst, right? So 8 to 32 celled blastocyst. And where does this blastocyst get implanted? It gets implanted in the uterus. So even during in vitro fertilization, when the 16-celled blastocyst is formed, it will be transferred where? To the uterus, so that it gets implanted easily and undergoes development in the uterus. Several hormones like HCG, HPL, estrogen, progesterone are produced by. Your main hint over here is this. So HCG is what? Human chorionic gonadotropin, isn't it? And HPL is what? Human placental lactogen. Yeah, what is it called? It's called human placental lactogen. So what comes over here? Placenta, right? So hormones like HCG, human placental lactogen, estrogen, progesterone are secreted by the placenta. Placenta is a temporary endocrine gland. What is placenta? Placenta is a temporary endocrine gland. Are you getting it? And its function is what? Its function is nutrition, because it provides nutrition to the developing embryo. It gives the oxygen, glucose, etc. to the developing fetus, developing embryo. Okay, nutrition. Then its function is what? Respiratory. Why respiratory? Because it's through the placenta that the embryo gets oxygen. Okay, oxygen. Then when oxygen is provided to the embryo, and then carbon dioxide is also removed through the placenta itself. So nutrition, respiration, then excretory. The nitrogenous wastes that are produced by the fetus are also transported through the placenta to the mother. And then endocrine. Yep. So endocrine, why? Because it is secreting hormones. So what hormones is it secreting? Human chorionic gonadotropin, human placental lactogen, yes, which induces lactation, estrogen, progesterone. These are secreted by the placenta. Got it? If a colorblind man marries a woman who is homozygous for normal vision, the probability of their son being colorblind is, let's do that. So capital C, normal, small c, colorblind. Yeah. So what does it say? Colorblind man. So X, small c, Y marries a woman who is homozygous for normal vision. So that is crossed with capital C, isn't it? Normal condition. So let's make a quick Punnett square. Yep. What is the question? What is the probability of their son being colorblind? So there are 0%, isn't it? Yeah. The son that will be formed over here will not be colorblind at all. whereas the females will be normal phenotype but carrier genotype. Yep. Now, so these females will be, this female will be now capable of transferring the disorder to her son. Understood this? So remember, with X-linked inheritance, this is X-linked, isn't it? Colorblindness is a sex-linked disorder. But which one? X-linked, and it is a recessive disorder. So it can be seen only in homozygous condition, but where? In females. In males, since there is no other X chromosome to overcome the defective allele on the X chromosome, the hemizygous condition, like if this particular hemizygous condition with a defective allele can still cause color blindness. Okay, so color blindness is a sex-linked disorder, and what does it follow? It follows crisscross inheritance. Okay, that is from male to female to male and so on. Okay, so that is what is crisscross inheritance. So the probability will be 0. Yeah. Next, in Hardy-Weinberg equation, the frequency of heterozygous individual is represented by, okay, so Hardy-Weinberg equation basically tells us, gives us ideal condition of evolution not happening. So it is dependent on the assumption that the frequency of alleles changes, frequency of alleles remains constant. Okay, it does not change at all. So the reasons like mutations, genetic drift, gene flow, all these affect the Hardy-Weinberg equilibrium. Yeah, and the equation that is given is p plus q the whole square, which is 1. So which is p square plus 2pq plus q square equals to 1. Yeah, it's supposed to be constant. Now p is the frequency of the dominant allele, q is the frequency of the recessive allele. So p square gives the frequency of the homozygous dominant condition, q square gives the frequency of the homozygous recessive condition, and 2pq gives the frequency of the heterozygous condition. Yeah, so this is how Hardy-Weinberg equation or Hardy-Weinberg equilibrium works. Yep. So that was with the questions for today. Yeah. If you have any doubts, you can put them in the comment section and we will take them. Yep, we will answer the doubts. That's done. Yep. Nothing? Great. So have a nice day, guys. Keep studying this.